Hello dear students. In this video, we're going to get general information about biotechnology and genetic engineering. Let's start. What is biotechnology? The term biotechnology, sometimes shortened to biotech, consists of two parts. Bio is the Greek word for life, and technology gives an indication of human intervention. Biotechnology can be based on the pure biological sciences, genetics, microbiology, animal cell culture, molecular biology, biochemistry, embryology, cell biology. Also, its interests can be outside the sphere of biology, chemical engineering, bioprocess engineering, information technology, biorobotics. Biotechnology deals with brewing, manufacture of human insulin, interferon, and human growth hormone, medical diagnostics, cell cloning, and reproductive cloning, the genetic modification of crops, bioconversion of organic waste, and the use of genetically altered bacteria in the cleanup of oil spills, stem cell research, and much more. As a matter of fact, biotechnology is very ancient. 6,000 years ago, microorganisms were used to brew beers and to produce wine, bread, and cheese. Yeast makes dough rise and converts sugars into alcohol. Lactic acid bacteria in milk create cheese and yogurt. This application of biotechnology is the directed use of organisms for the manufacture of organic products. Examples include beer and milk products. In this way, classical biotechnology refers to the traditional techniques used to breed animals and plants, as well as to the application of bacteria, yeasts, and molds to make bread or cheese. Modern biotechnology came into being during the 1970s. It has often been divided into several categories. Every field of this science is sometimes connected with a definite color. Green biotechnology is biotechnology applied to agricultural processes. An example would be the selection and domestication of plants via micropropagation. Another example is the designing of transgenic plants to grow under specific environments in the presence of chemicals. One hope is that green biotechnology might produce more environmentally friendly solutions than traditional industrial agriculture, although this is still a topic of considerable debate. Red biotechnology is applied to medical processes. Some examples are the designing of organisms to produce antibiotics and the engineering of genetic cures through genetic manipulation. White biotechnology, also known as industrial biotechnology, is applied to industrial processes. An example is using naturally present bacteria by the mining industry in bioleaching. So it is a designing of an organism to produce a useful chemical or destroy hazardous, polluting chemicals. Blue biotechnology is a term that has been used to describe the marine and aquatic applications of biotechnology, but its use is relatively rare. Genetic engineering is the artificial manipulation, modification, and recombination of DNA or other nucleic acid molecules in order to modify an organism or population of an organism. The term genetic engineering is generally used to refer to methods of recombinant DNA technology, which emerged from basic research in microbial genetics. The techniques employed in genetic engineering have led to the production of medically important products, including human insulin, human growth hormone, and hepatitis B vaccine, as well as to the development of genetically modified organisms, such as disease-resistant plants. Historical developments. The term genetic engineering initially referred to various techniques used for the modification or manipulation of organisms through the processes of heredity and reproduction. As such, the term embraced both artificial selection and all the interventions of biomedical techniques, among them artificial insemination, in vitro fertilization, for example, test tube babies, cloning, and gene manipulation. In the latter part of the 20th century, however, the term came to refer more specifically to methods of recombinant DNA technology, or gene cloning, in which DNA molecules from two or more sources are combined either within cells or in vitro, and are then inserted into host organisms in which they are able to propagate. Applications. Genetic engineering has advanced the understanding of many theoretical and practical aspects of gene function and organization. Through recombinant DNA techniques, bacteria have been created that are capable of synthesizing human insulin, human growth hormone, alpha interferon, a hepatitis B vaccine, and other medically useful substances. Plants may be genetically adjusted to enable them to fix nitrogen, and genetic diseases can possibly be corrected by replacing dysfunctional genes with normally functioning genes. That's the end of our video session. Next time we're going to speak about characteristics and classification of living organisms. Thank you for your attention.